Hello, welcome to today's Coffee Science Seminar. If I can have the next slide, please, Patrick.
Oh, hang on. Is it working?
Not yet.
There we go. There we go.
So today's seminar will be by Patrick Mason. The next slide, I think, indicates that I'm Robert Henry and I'll be facilitating today's seminar. The next slide, Patrick. I'd like to start by acknowledgement of country. The University of Queensland acknowledges the traditional owners and their custodianship of the lands on which we meet, and that's the very many places on which those participating in the seminar today will be. We pay our respects to their ancestors, their descendants, who continue cultural and spiritual connections to country, and we recognise the valuable contributions to Australian and global society. Next one, Patrick. So just a bit of housekeeping. Seminar goes from 12 till 1. After the presentation, we'll hold a question and answer session. And throughout the webinar, please put your questions into the Q&A tab, not the chat function, and we can then address those in the question and answer session. So it's over to you, Patrick. Patrick,
awesome. Thank you.
I'll just introduce you, sorry. Patrick is a postdoc with Coffee. He works from the molecular to the field level, very diverse range of projects in plants, working, I think, in horticulture and crop science projects that really include examples of food crops, industrial crops, and pharmaceutical crops. So Patrick's someone with very broad experience in this area. So I'll hand over to him to talk to you about his sorghum mutant work.
Fantastic. Thank you very much, Robert. And I'd also like to acknowledge the traditional owners and their custodianship of the lands in which we meet today. So today I'm talking about the generation of a large-scale mutant library in sorghum that we completed last year. And just to quickly go through what I'm going to be talking about today, basically just be going through the two phases of the project. So the first phase was sort of optimizing an EMS treatment protocol and also figuring out how many mutants we actually needed to. I mean, the aim really is to get a knockout in every single gene. So if the mutant population is big enough, that way you can get a mutation pretty much in every gene, or sorry, a knockout in every gene. And then moving on to phase 2, which was actually generating and applying this optimized treatment protocol, and then generating the large-scale mutant library, and then screening that mutant library for particular genes and mutations in those genes. So where did all this start? Well, this work and the development of this mutant population is funded by the Carlsberg Research Laboratory. And it's centered around the use of this particular protocol. It's called the FIND-IT protocol. And it was developed by Søren Knudsen and the team at Carlsberg. And this was in barley, which makes perfect sense, seeing as though they are a beer company and it's sort of central to beer production. And basically, this technique, it's quite similar to tilling in many ways, but the main advantage that it has is that you can screen enormous numbers of mutants in a very short amount of time, and get to the point where you actually isolate a particular individual that will have a knockout of your specific gene. So the idea is that if you produce enough mutants, you can end up having most likely a knockout in every single gene, and that, I guess, has a lot of value from a breeding perspective. So you can actually specifically target knockouts of genes that you're looking for. And so I guess this is sort of an alternative to GM, really. The rules are a bit wishy-washy these days, and I know that maybe we'll move beyond it and we'll be able to use GM more so than we are right now. But until that day, this is definitely one of those options that you can use to circumvent that. And on top of that, sorry, I've lost my train of thought. But anyway, I'll just quickly go through the generation of the library. So basically, you take the seed, you treat it with your particular mutagen, and you sow those seeds in the ground, and then you harvest the seeds from those particular plants, and then you sow the M1 population. And then from there, you actually harvest those seeds, but you collect them into bulks of seed, as in 300 seed head bulks. So in this case here, in this barley population, they produce 500,000 individuals, of which those 500,000 individuals were bulked into 300 seed head bulks. So that turned out to be about 1,600 bulks. And from here, you actually took 25% of the seed from each of those bulks, you homogenized it, and you extracted the DNA from that. And then so from each of these bulks, you extracted the DNA, and then you ran it through digital droplet PCR, which essentially allows you to find a particular mutation in your gene of interest using TACMan primers. And so from there, so once you actually find which specific pool has your particular has a mutation or a knockout in your particular gene of interest, then you move through to phase 2, by which so you've got your particular bulk, which is your 300 seed head bulk, and then from there you do sort of subpools or subpool bulks, wherein you take around 1,000 grains, and you take flour from 10 seeds each and have them in mini bulks. And so you take flour from those 10 seeds, you place them into a 96-well plate, into individual tubes, and in a corresponding 96-well plate, you retain your seeds. So if A1, you put your flour sample in an A1, and then you put all your seed in a corresponding A1. So you know that if you find your particular gene of interest in that particular well, then you know that you need to grow out those particular seeds to find the individual that contains that mutation. And then from there, you can either run that on digital droplet PCR or qPCR in exactly the same way that you've done the initial screening with digital droplet PCR. And then from there, once you've found the 10 seeds that contain your particular mutation, then you move to phase 3, where you actually grow the plants up, and you take a small cutting from each of those plants, and then from there you run the PCR again, and then you can actually find the individual that actually has the particular knockout you're looking for. And so again, the main time-consuming factor of this is the production of the library and the DNA extraction. Once you've got the DNA extraction of the actual pools or the 300 seed head bulks, once you've done that, everything else is very quick. You can find your particular mutation of interest in your particular gene in less than a week. It's very, very quick once all of that groundwork has been done. And so this has a particular application over, say, CRISPR knockouts, because you need quite a bit of infrastructure to actually get that going, and it costs quite a bit of money, whereas this is probably a more accessible option for a lot of different groups around the world who don't actually have that capacity. So why sorghum? Why is Carlsberg funding a project in sorghum, and why are we doing it? Well, it's a very important crop, and it's probably going to become a more important crop as we move into a world that's more and more affected by climate change. So it's a C4 plant. It can deal with high temperatures. It does require less water, and it's more drought tolerant than most other cereal crops. So we may actually see this crop being utilized in certain areas where traditional cereal crops were utilized. And on top of that, it's a very versatile crop as well. So it is used in a variety of cultures and countries. It's used in the human diet, but also as livestock feed. From an alcoholic perspective, make spirits with it, but also from a beer making perspective, if we're looking at Carlsberg, it can actually be a grain additive in beer production as well. And then more recently, with the really inflated cost of fossil fuels, there really is an interest in utilising sorghum as a biofuel crop, particularly in that fibre fraction. So mutagenesis, it's really the basis of species evolution, and it's something that plant breeders have been using forever, really, to select for novel traits, and there's mostly in a natural context, but more recently, say in the last 80 to 100 years, people have been using, or researchers and breeders have been using chemical, biological, and physical mutagens to produce mutants that have novel traits. So in the context of sorghum, it is limited by a very narrow genetic base, and there is quite a bit of diversity in the sorghum genus. However, they do have a number of desirable, undesirable traits, as do a number of non-commercial varieties as well. And one way to avoid having to worry about these undesirable traits, you can actually take a very high quality line that you would use in a commercial context and mutagenize those individuals, and it reduces the need to get rid of those undesirable traits. And so one of the most commonly utilized mutagens in sorghum is ethyl methanesulphonate, and this was chosen for our project. And in terms of what you can get out of this, a great example of a gene that's derived from EMS mutagenesis is a brown midrib gene. And so this is actually a knockout of one of the CAD genes, which is associated with, or which is part of the lignin synthesis pathway. And by knocking out this gene, you actually end up with the cell which has quite a bit less lignin, which makes it less recalcitrant to utilizing that cell wall for a number of different purposes, but particularly from a bioproduct or biofuel perspective. So that's an example of how valuable an EMS mutant can actually be from a commercial perspective. And so just to quickly run through what EMS actually does. So it causes random point mutations. So it's not causing huge frame shifts or frame shifts. It's just causing a single base pair mutation, and that's in the guanine base. So basically what happens is this results in the formation of ethyl guanine, and the mutated base pairs with thymine instead of cytosine. And so what you end up getting is you start up with your CG in your original wild type sequence, and what it will turn into if the EMS affects that guanine, it turns into a TA base pairing. And this can lead to a number of different things, particularly in the coding region, specifically stop codon, like the formation of premature stop codons, or missense variations as well. So what are the aims of the project? As I said before, we're developing an optimized protocol for EMS treatment of sorghum in the context of the funded protocol. So really, we want to figure out how many mutations we're getting depending on the rate of EMS we're using, and also how many mutants do we actually need to actually get or likely get a mutation at every single gene in the sorghum genome. Sorry, not a mutation, a knockout in every single gene in the sorghum genome. And so, so we'll be subjecting the sorghum seed to different rates of EMS, and we'll be testing that using NGS and looking at the field attributes as well of the treated plants. And then phase 2 is using this optimized EMS protocol and produce a very large population of sorghum mutants that can be screened for knockouts. So phase 1. So back in early 2021, we started this at very short notice. And really, again, it's about analyzing how EMS affects sorghum from a physical and genomic perspective. And so we ended up treating the seed at 4 different EMS concentrations. So we had a control, 0.05%, 0.15 and 0.25% EMS rates. And these rates were based around a rate that was used by Celia Blomstead back in 2012 in sorghum, and she produced a population of 6,000 mutants, and it worked quite well. So we decided we'd base our study around that rate. So we'd have a higher and a lower and a rate that was the same as that. So yes, we treated the sorghum seed with EMS, and so this seed was single seed descent inbred BT623. And this is particularly important because this is what the reference genome is as well, which makes it a lot easier to analyze from a genomic perspective. And then this seed was sown and field observations were made, and then the seed was harvested. And so we're able to collect a number of different data points from the field. And then from there, we took the M1 seed and planted it in the glasshouse, and we collected tissue from the M2 plants for DNA extraction. And so these M2 plants were representatives of all the different EMS concentrations. And then we sequenced that with Illumina whole genome sequencing at around 15x coverage, so which is plenty for this type of application. So we'll be doing a variant analysis, which was done in CLC. And yeah, so yeah, this is just an overview of the field. So just to start things off, it's analysis of the field data for figures A and B. We didn't see any significant difference between the treatments. I think particularly with figure A, it's probably because we didn't have enough data points. I think if we had more data points, we probably would see a significant difference in field survival. However, there was absolutely no significant difference at all, or any observable difference in figure B, which is in terms of pest occurrence. But the big thing here is the mutant height, which is figure C, and you can and what you can see is there is a significant difference. So pretty much as you increase your EMS rate, the lower the mutant height. So it is having quite a significant effect on the growth rate and the growth habit of the plants that are affected by EMS. So the more EMS you add, the less growth you're going to have in a plant. And then moving through to field, to the field and seed related data. So figure A shows just the basic seed yield. So how much seed were we getting per head based on the EMS rate? And as you can see here, the more you add EMS, as you would expect, there's going to be less. You're going to get less seed off of those plants, and there's quite a precipitous drop in seed yield between the 0.5, the 0.05 and the 0.15 EMS treatments, and at the 0.25 EMS treatment, there's next to no seed on the seed heads at all. And if you move over to figure B, you can just see how significant an effect the EMS treatment has on the amount of seed you get on a head. So you can look here that let's just look at the 0.25 EMS treatment. The percentage of heads with less than 1 gram of seed was around almost 70%, and then on top of that, there was about 30% of those seeds were seeds with 0 grams of seed. So about 95% of those seeds in that treatment group had next to no seed on it, and that's really significant as well, because when you're screening with the find it protocol, again, it's the more seed you have, the better the chances you're going to find that mutation of interest. So there is a point of diminishing returns. You can hit it really hard with EMS, and you are going to get more mutations most likely. But in this case, you'll end up with next to no seed. So it's definitely something to weigh up. And then moving over to seed germination, which again is also quite important for the find it protocol, because when you move through to that phase 3 and you're germinating those 10 seeds that you found, the less that germinate. So if it's 8 that germinate in a lower EMS treatment versus say around 6 or 7, that's significant, because those 3 that don't germinate might actually be your mutation of interest. So it is very important to try and also maximize that seed germination rate. And there were significant differences between the 0.5, the 0.05 and the 0.15 and the 0.25 treatments. And here's just an example of the effect that EMS has on the seed heads. And there is quite a bit of variability there. So the seed head on the left here, that's got about 80% seed head coverage, and the seed yield is probably around 20 grams, versus this one here, which is probably less than a gram. I think there's a couple of seeds in there. So there's quite a bit of variability. And I think this is actually within the 0.15% treatment group. So you can actually get individuals that will get quite a few seeds on the head, but most of them will actually be blasted, and there'll be literally no seeds on the head. So that's, again, something to be made note of, and something to be taken into account. And so moving on to the sequencing and variant analysis. And so these are the plants that we collected the DNA from. And from this, we produced 30 Illumina whole genome sequencing libraries. And again, we got around 15 to 20x coverage. And you can see here that we have our control group, and we did 10 samples from this control group because we wanted to build a background variant library, because there are going to be some significant differences between our seed, our BT623 seed, and the genome that was produced 6-7 years ago as well. So there's going to be quite a bit of difference between those individuals. So it's important to filter out all of those background variants, so you know that all you're looking at is EMS-based, EMS-derived variants in your treatment samples. And so these are just this is just an illustration of the addition of those control samples, and it shows how important it is in terms of having higher numbers of control samples to actually filter out all these background variants. And so you can see here that the more you add, the more control samples you add to the background variants, the more of those background variants you actually are able to filter out and remove and find. And so and just one other thing to note, and this is from the background variant library that we created. What I did note is that there's a significant difference in mutation rate depending on the region of the genome that you're in. So if you're looking at a whole genome level or in the intergenic region, the mutation rates are very high. But if you look in the regions that really matter in terms of the survivability of a plant, so the coding region or the genic region, the mutation rate is significantly lower, and that's quite interesting. And so there's significantly less mutations in those regions, and which is quite important. And it's probably good in our sense, because I think we need a lot more control sequencing. We need to sequence a lot more control samples to actually find and get all of those background variants. However, in the coding region, because that number is so much less, it's very likely that we've gotten 99% of those variants in the coding region, because there's so few mutants in those areas. And so, yeah, like I said, those 10 control samples used to make a database to filter out background variants. And so the question would be, is the background filtering working? So as I noted, there were a lot of background variants at a whole genome level. And just to quickly explain what these figures actually mean. This is the nucleotide transition type. So this GC to AT transition is what the EMS mutants mutagenesis will actually cause. However, there's already a lot of this. There's already a lot of these mutants already in the genome. So it's important that we try to filter out as many of those as possible. So we know which ones are actually produced by the EMS treatment. And what you can see here is that using the background filtering against the sequencing that we did in the EMS treated samples, you can see that it is quite effective at a whole genome level to removing most of those mutations in these treated samples. However, quite a few are carried through. So you can see here on the 0.05. 25% are still carried through, and so these are others. So we know these aren't EMS derived. So there's a bit of carry through there, and that is important to note. So we need to make sure that we're not counting stuff that aren't mutants that aren't genuine EMS derived variants that weren't already in the genome as it was. However, if you look in the coding region, there's a significant difference here. And as I said before, there's significantly less variance in the coding region. So as you can see here, In the control, we see the same sort of percentage in terms of EMS type variants that were found in the background variants. And so it's similar to what you see at a whole genome level. However, when you filter just the coding region in these EMS treated samples, you see that we're able to filter out pretty much all of the other samples. So we can have a pretty high degree of confidence that in the coding region, which for us and for the find it protocol really is the region that mostly matters, it's good, and I think it'll be fine for the rest of our analysis. And so, as I said, 98% EMS type variants and only 2% other background variants carried through. So that's a pretty good result. So quick look at the variant analysis across a whole genome and how many variants you'll actually see based on the EMS treatment. And as you can see, at the lower EMS treatment at 0.05, significantly lower, around 2,500 variants across the genome. Whereas if you up that rate to 0.15, we're looking at around 6,000 EMS type variants. And then it goes up again at 0.25. However, I don't think we have enough data points to really resolve that, because there is quite a bit of variability because each seed, even if you're mixing it quite well during the treatment process, is going to get a different dose of EMS. So there is going to be quite a bit of variability between each individual, which is understandable. And one thing to note again is that as we saw in those background variants, also in EMS-derived mutations as well, the mutation rate in the genic and coding regions is also significantly lower. So there is a degree of selection going on there. So individuals that have a lower amount of mutations in the genic and coding regions are more likely to survive than ones that have more, which makes perfect sense. And so here really this is the coding region. This is the amount of variance in the coding region. This is the area that really matters to us and what we're sort of what you'd be screening for in the find it protocol. And so this is synonymous, missense, and premature stop codons, all in the coding region, obviously. And you can see here that, yeah, once again, seeing that same trend of significantly lower amounts of mutations in the 0.5, and then the 0.15 and 0.25. There aren't any real clear differences. There was actually, I did have significance on there, but there's a typo here, so I do apologize for that. So anyway, to focus in on these premature stop codons, really this is what, during the find it protocol, when designing primers, this is what you're designing them for. You're designing them for premature stop codons. And then if you can't find that, then you start aiming for the missense mutations, because premature stop codons are the most likely to produce a knockout in your particular gene. And so I did want to investigate this further. So from those 20 samples that we did sequence that were from the EMS mutant population, I did want to see what these genes were where stop codons were produced, and what it actually looked like, and if they would be actually a successful or show an example of a successful knockout. So this is just an annotation that was done in omics box, which shows the identity of these 180 genes that we found premature stop codons formed as a result of EMS in those 20 samples. And just focusing in on one particular gene. Now, I don't really have any interest in galactose metabolism at all, but this was something that I just wanted to highlight, where so you have this particular gene, and let's just say you are interested in galactose metabolism, and you're searching for this gene in the find it protocol. This is an example of a situation or a mutant that would very likely cause a knockout. So using the annotation, you can find where in the actual gene the knockout actually is, or sorry, the stop codon actually is, and you can see here it's in the first third of the coding sequence. And this is most likely a knockout. So this is a perfect example of where you design a probe, and there are programs for this that you can use that. But this is a perfect example of something that would actually produce a knockout in this particular gene. So if you're interested in that, then you've got a knockout, and you can investigate what type of phenotypes you would actually get from a knockout of this type of gene. So moving on and sort of concluding the phase one. Which EMS treatment concentration is optimal for producing variants? So as you saw before, the more EMS you treat with, you're going to get more variance in the coding region. But the problem is your seed yield is going to go down. So yes, you're going to get more mutants, but you're also going to get more. You're going to get less seed, and so the likelihood that you're going to get a huge diversity of individuals that you're getting seed from is significantly lower. So from my understanding, I think this is probably the best way to think about it. I think there's a precipitous drop there in seed yield, but there's also a precipitous increase in the amount of variance that you get. So somewhere in the middle here is probably an optimal EMS treatment concentration. And so moving on to figuring out how many mutants do you actually need to get a mutation at every gene. Now, this is a very rudimentary calculation, and so I've basically tried to figure out how many mutants you would need to get a mutation at every possible base. And I know that this isn't how probability works, but it is a good illustration of how different the numbers you need based on the variant count you get based on the EMS treatment you do. And you can see there's a huge difference between a 0.05, which is quite a light treatment of EMS, and 0.15 and 0.25. And it seems to me like between 0.15 and 0.25, there's diminishing returns. So there's no real reason to increase your EMS dosage beyond 0.15 because there are diminishing returns. However, there is a sweet spot between 0.05 and 0.15. And so it's about figuring out how many you actually need and how best to do that. But in this particular circumstance, there is such a disparity between 0.05 and 0.15. So summary of results, the most important parameters that dictate an optimal EMS rate would be coding region variant counts, seed yield, and percentage of seed heads without seed. And then based on these parameters, we think that the best EMS rate is definitely between that 0.05 and 0.15% range. So probably maybe just in the middle, 0.1 would probably be the best. And the number of mutants required to produce a mutation at every base is greatly affected by EMS rate. So particularly between 0.05 and 0.15%. So if you don't have the resources to bear to produce a very, very large population, and there's a lot of work involved in collecting and also extracting the DNA, it might be better just to increase that dosage in this range a bit, so you can reduce the amount of mutants that you actually need to produce. So moving on to phase 2, the generation of the mutant population screening. So this is a picture of the field, and yeah, it was quite a lot. It was a lot of work, but in the end, it was successful. But and so just to quickly go through what we're doing based on the finite method that we're going to utilize in sorghum. So this is a figure that was produced by Anko at Carlsberg. So he's been involved in screening the entire population that we've produced. And basically, it's a little bit different to what we saw in barley. There isn't 2 generations that are grown out of the mutants. We start at the M1 generation, and we collect most of the grains at this M1 stage. We did have some M2 plants that we did produce during the pilot scale study, but it was a smaller population. So most of the plants that we did produce were M1 plants. And everything else is pretty well the same, other than the bulks that we collected. Instead of 300 seed head bulks, we produced, we collected 100 seed head bulks. And so this is the seed treatment. So we treated with 0.1% EMS for most of the treatment runs. However, we did end up producing some treatments that were 0.15%. We were worried about making sure we had a number of mutants that had a slightly higher dosage to ensure that we got a knockout in every gene. However, these particular runs that we did produce significantly less seed, so that has to be taken into account as well. And so yeah, we ended up planting 8 hectares of M0, but also M1 seed that was derived from the pilot study as well, which was a smaller amount. And this is just an image of the tractor that we used. So it was a long process, and it was pretty hot in the back of the tractor, but we ended up getting it done, so it's all good. So harvest. So what harvest looked like is you pretty much walk around with a Hessian bag, and you would collect 100 seed heads by hand and put them in that Hessian bag label it, and then tie it off and put it in the back of the trailer. Take it to an oven, dry it off at 40 degrees for about 3 to 4 days, and then from there we'd thresh it. And from there we'd thresh, and we'd split each 100 seed head bulk into two, and half of that bulk we sent to Carlsberg, and half of that bulk we ended up keeping here in Australia for long-term storage. And as you can see here, we ended up producing 160,000 mutants, which is 1,600 bulks in the end. So just a quick overview of the screening process of the sorghum. So yeah, we shipped the grains to Carlsberg Research Lab in Denmark, and 25% of that half bulk was milled, and then DNA was extracted from that and then screened with digital droplet PCR. And this is an example on the digital droplet PCR of a positive hit. You can see all these blue dots here. And that shows you which specific bulk actually has that mutation of interest that has a hit on the TACMAN probe. So and then from there you move to phase 2, and you're drilling 95 times 10 grains, so 10 grams 95 times, and you're putting that into your 96 well plate, and then you're having one control well. And you can see here, you collect the flour, and that goes to the DNA extraction process. But on the left here, you can see these small blue boxes. This is the corresponding well had the 10 seeds put into it. So again, if you get a hit on well B4, then you can say, okay, well, these 10 seeds definitely have the knockout in my gene of interest. And then from there, you go through to growing out those 10 particular plants. Oh, sorry, yeah, doing the drilling, and then doing the qPCR or ddPCR again. And then from there, once you've found that individual well, then you grow out those 10 plants, and from there, then you can find the actual individual that actually has that knockout of interest in that gene of interest. So, summary of this. So we've created a mutant library consisting of 165,000 M1 and M2 plants. This is completed midway through 2022. And so half of the seed has been sent to Copenhagen and half has been kept in Australia. And so far, Carlsberg has extracted the DNA from 2/3 of the bulks that we've sent. And even though they've only done that for 2/3 of the bulks, they've been successful in isolating 29 out of 31 of the gene targets so far. So the strike rate's been very, very good. And I think that if they redesign those primers as well for those 2 genes that they didn't find, they'll most likely find they'll end up finding those knockouts of those particular genes. And so at the moment, we're trying to apply the find it method from phase 2 in Australia. And yeah, it's still a work in progress, but we're getting there. And I think we're pretty close. So what basically Denmark will do, or the Carlsberg Research Lab will do phase 1, which is with a digital droplet PCR, and then they'll identify the bulk for us. And because we've got half here, we can take that bulk, do the seed drilling, and do qPCR on those individual wells and find those particular individuals. And so moving on to future work, apply the phase 2 protocol at UQ, and then identify and grow out those mutations containing our mutagens of interest, because we've got some targets that we've had screened in Denmark, and we're doing that here now. And we're hoping to utilize those mutants in Robert's upcoming project, which is an ARC hub to replace fossil fuels with plants. And just a few acknowledgements from the Carlsberg Research Lab, the group there, particularly Anko, Soren. Anko's done a lot of work, a huge amount of work in the screening process. So it's been extremely helpful in helping us get our phase 2 find it protocol off the ground. Also like to acknowledge Agnello as well, because he helped me a lot with the mutant or the variant screening process. And then Peter as well, he guided me throughout the entirety of the field work as well. It was huge population, a lot of work, managing a lot of people for the first time, and he was always there, so he massive help from him. Mia, his assistant, and Rajesh, he was also there a fair bit of the way. Biwa and the group at University of Copenhagen, the fieldwork team at Gatton, and Ha, she's been doing a lot of seed drilling at the moment, trying to get that qPCR here off the ground. Robert Henry and the Associated Lab Group, and also Travis Dance and the Crop Research Unit team. So big thank you to all. And yeah, that's it. Questions? Robert, I'll hand it over to you so you can facilitate this. I can't remember what to say here.
Thanks, Patrick. We're now certainly open for questions. So please put your questions in the Q&A section. I might start, Patrick, by while we're waiting to see if there's any questions there, by asking you about the genes that you're failing to find. 29 out of 31 targets that people wanted to find mutants in were successful. Is it possible that the other two you're not finding because they're lethal, or is it that you just haven't screened enough?
It's possible that it is possible that they're lethal. And I think Anko did intimate that, and I actually forgot that. So thanks for reminding me, Robert. But yes, you're absolutely right. Yes, that's definitely the case. It could be the case, yeah.
And can you comment on the extent to which you're finding homozygous versus heterozygous mutations with this strategy only at M1?
Yes. Well, I guess you're getting a lot of seed. It just depends on how much seed you have. If you've only got a few individuals, then less chance you'll find a homozygous seed. But yes, there have been a number of homozygous mutants found with the particular gene of interest, but I think most of them, I believe, were heterozygous. So you're going to have to go through a process of growing another plant out, and hopefully you'll get some homozygous hits for those particular mutations.
Thanks. So I've got a question here. My question is, are you also looking for already available mutants in UQ's sorghum population? Not sure what the answer to that would be.
What's the question, sir?
My question is, are you also looking for already available mutants in UQ's sorghum population?
Well, I think it belongs to. I think we've got bulks. So we split the bulks in half. So we actually have we're sharing the bulk. So we're able to find the same mutants in our bulks as they have in Denmark. So they're all available. And yes, so I guess in answer to your question, yes, we are, and they've already identified mutants and have isolated them in this population, but it belongs to UQ and Carlsberg, or Carlsberg specifically, sorry.
Patrick, another question. Gita, great talk. Just a question regarding the lignin biosynthesis gene CAD. You were successful in knocking out this gene. Was there any compensation or by any other genes?
So we didn't do that work. This knockout was found quite a number of years ago. It was probably over a decade ago, actually. I don't remember the paper. It was a specific isoform of CAD. I don't know if you remember, Robert, but it's a particular isoform, and knocking it out, it must be the key in that pathway, and it really has a major effect on the amount of lignin that gets deposited in the cell wall. And if you're asking if we have actually knocked out this gene, we probably have. I dare say we have. Our population is big enough, but we haven't screened for it yet. But I do think it'd be worthwhile screening for it, just to maybe validate the screening procedure and also phenotyping.
Okay, another question. Gamma ray irradiation is an alternative method of mutagenesis. Do you think gamma radiation is as effective as EMS? And what level gamma radiation is comparable to the 0.1% EMS treatment?
I'll be honest, I couldn't for the life of you tell you what gamma radiation actually does. As in the terms of what type of mutations it actually causes. So I know people use it, and I think UQ offers that service, irradiation, but I haven't read up enough on it, so I do apologize. I don't have an answer for that.
I can just add, it's not a way of generating point mutations. It's usually used to knock out whole sections of the genome. So it's not the sort of precise tool that EMS can be. Secondly, is there any specific trait mutagenesis you will focus on after screening, or will you go with the investigation of all the mutants? Maybe you need to explain that.
Yeah, so it'll be centered around our particular focus. I mean, a lot of the focus at Carlsberg has been for different things, centered around the Duran pathway, but for us, it was centered around biofuel traits. And so that's what we're screening for at the moment, and that will be a part of a small part of Robert's hub. And in terms of will this project lead to stable mutants? Well, it's been successful in barley. We haven't gotten to the point where we've produced stable mutants yet, but we're pretty confident because it has been successful in barley that it will be also successful in sorghum.
Okay, so that's a good range of questions. Do we have any others? It looks like you might have answered most people's questions, Patrick. I think if there aren't further questions, we still have a little bit of time, but you might take the opportunity to wrap it up there.
Perfect.
Thank you, Patrick. Can we just go on to the next slide?
Absolutely.
Just to announce that the next seminar in this series will be on the 17th of October, Arun Kumar, on the role of phytase enzyme in monogastric nutrition. So I hope you're all able to attend the next seminar. Thanks, everyone.
Thank you very much.
Bye, everyone. Thanks.